The discipline of the discipline of medicinal chemistry is devoted to discovery and develop of new agents for treating disease. Most of these activities is directed to new natural or synthetic organic compound, paralleling paralleling the development paralleling the development of medicinal agent has come a better understanding of the chemistry of the receptor. The latter has been greatly facilitated by low-cost computer running software that calculate molecular property and structure and pictures it using high-resolution graphic. Development of organic compounds has grown beyond traditional synthetic medicine method. It now includes the exciting field of biotechnology, using the cell biochemistry to synthesize new compound. Technique ranging from recombinant DNA and site-directed mutagenesis to fusion of cell line have greatly broadened the possibility of new entities that treat disease. Pharmacists now dispense modified human insulin that provide more convenient dosing schedules, cell-stimulating factor that have changing the dosing regimen for chemotherapy, humanized monoclonal antibodies that target specific tissue and fused receptors that intercept immune cell generate cytokinin. This 12th edition of Wilson and Giswold textbook of organic medicine and pharmaceutical chemistry continues the philosophy of presenting the scientific basis of medicinal chemistry, originally established by Professor Charles Wilson and Oli Giswold, describing the many aspects of organic medicinals, how they discover, how they act, and how they develop into clinical agent. The process of establishing a new pharmaceutical is exceedingly complex and involve the talent of people from various disciplines including chemistry, biochemistry, molecular biology, physiology, pharmacology, pharmaceutics, and medicines. Medicinal chemistry itself is concerned mainly with the organic, analytical, and biochemical aspect of this process, but the chemist must interact productively with those in other disciplines. Thus, medicinal chemistry occur occupy a strategic position at the interface of chemistry and biology. All the principles discussed in this book are based on fundamental organic chemistry, physical chemistry, and biochemistry. To provide an understanding of the principle of medicinal chemistry, it is necessary to consider the physicochemical property used to develop new pharmacological activities, compound, and their mechanism of action, and drug metabolism, including possible biological activities of metabolites. The importance of stereochemistry in drug design and the method used to determine what space a drug occupies. The earliest drug discovery were made by random sampling of higher plant. Some of the sampling although based on anecdotal evidence led to use the such crude plant drug as opium, belladonna, and ephedrine that have been important of century. With the accidental discovery of penicillin came the screening of microorganisms and the large number of antibiotics from the bacterial and fungal source. Many of these antibiotics provide the prototypical structure that the medicinal chemistry modified to obtain antibacterial drug with better therapeutic profile. With the changes in federal legislation reducing the efficiency requirement for nutraceutical, the public increasingly is use so-called non-traditional or alternative medicine that are sold over the counter. Many outside of traditional pharmacy distribute channel. It is important for the pharmacist and the public to understand the rigor that is required for prescription only and Food and Drug Administration (FDA) approved non-prescriptional product to be approved relative to the non-traditional product. It is also important for all people in the healthcare field and the public to realize that whether these traditional products are effective as claimed or not. Many of the alternate medicines contain pharmacologically active agents that can potential or interfere with physician prescribed therapy. Hundreds of thousands of new organic chemicals are prepared annually throughout the world, and many of them are entered into pharmacological screen to determine whether they have useful biological activities. This process of random screen has been considered inefficient, but it has resulted in the identification of new lead compound whose structure have been optimized to produce clinical agent. Sometimes a legal develops by careful observation of the pharmacological behavior of the existing drug. The discovery the discovery that amantadine protect the treat early influenza. The discovery of amantadine the discovery of amantadine treat early influenza. It came from several screen of antiviral agent. Okay. Uh, Sometimes double careful observation pharmacological behavior existing drug. This use of amantadine in long-term care facilities show that it also could be used to treat Parkinson disorder. Parkinson disorder. More recently, automatic high-order throughout screening system utilizes cell culture system with link enzyme assay and receptor molecule derived from gene cloning have greatly increased the efficiency of random screening. It is now practical to screen enormous library of peptides and nucleic acid obtained from combinational chemistry procedure. Rational design, the opposite approach to high volume screen is also flourishing. Statistical method based on the correction of physicochemical property with biological potential are used to explain the optimized biological activity. Significant advantage in X-ray, crystallography, and nuclear medicine, magnetic resonance have made it possible to obtain detailed representation of the enzyme and other drug receptor. This technique of molecular graphic and computational chemistry have provided novel chemical structure that have led to new drug with potential medicinal activities. Development of human immunodeficiency virus, protect inhibitor and angiotensin converting enzyme inhibitor came from an understanding of geometry and chemical character of the respective enzyme active site. Even if the receptor structure is not known in detail, rational approach based on the physicochemical property of lead compound can provide new drug. For example, the development of cimetidine involve careful study of changes in angiotensin of H2-histamine receptor, including various physical property of structure based on histamine. As you proceed through the chapter, think of what problem the medicinal chemistry is trying to solve. Why were certain structures selected? What modifications are made to produce more focused activities or reduce adverse reaction or produce better pharmaceutical property? Was the prototypical molecular discovery from random screen, or did the medicinal chemist have a structural concept of the receptor, or understanding the disease process must be interrupted? Thanks for watching my chapter 1. Next our topic is chapter 2, drug design strategy. So friends, if you need this book, comment me on comment on this video. I will send this PDF book at free of cost.